right, so how you're doing today? How you're doing today? I'm doing good, thank you. All right, so we did stop last time at the abdominal pelvic regions, if you remember. What did we call those various regions of the abdomen? We divided the abdomen and pelvis together into 9 regions to be able to locate the exact site of pain, for example, the exact site of the complaint, for us to figure out what is the reason for this pain or the complaint. If you remember, we did have 3 middle regions. One is located above in the abdomen, epigastric. One is located below in the abdomen, hypogastric, and one in the middle. This is the umbilical region. On both sides, we've got the hypochondriac regions, right hypochondriac, left hypochondriac region, right lumbar, left lumbar region, right iliac, and left iliac regions. In order for us to discuss the internal structure of the body, we need to have sections in the body for us to see the internal structures. We have 3 main types of sections that we can perform in order to see the internal body structures. Those are the transverse section. And the transverse section here is going to be dividing the body into a superior part and an inferior part, upper and lower. A sagittal plan or a sagittal section is going to divide the body into a right part and the left part. The type of sagittal sections that passes through the midline is what we call the median section or the mid-sagittal section. So generally speaking, if I'm dividing the body into right and left parts, it's a sagittal. If the sagittal section is passing through the midline, we're going to call this as the mid-sagittal or the median section. The third type of sections that we're looking at, it's going to be the frontal section, which splits the body into an anterior part and a posterior part. Like here, the X-ray. When we have an X-ray done on the chest, for example, we can see a slice of the body that resulted from a frontal section, means you can see either the anterior or the posterior section through the slice. Unlike the MRI or the CT scan, we can perform transverse sections in which I've got 2 parts of the body, an anterior part, a superior part. This is superior, so this side is going to be the head of the patient, and on the other side is going to be the legs of the patient. In a sagittal plan, we're going to be cutting the body into right and left parts. If I'm passing through the midline, it's called median section or mid-sagittal section. When we look at the body cavities, we do have 2 main cavities in the body. Those were the dorsal or the posterior body cavity, and this includes the cranial cavity and the vertebral cavity. So dorsal body cavity is divided into two main cavities, the cranial cavity and the vertebral cavity. The cranial cavity is where you keep the brain. The vertebral cavity is where I keep the spinal cord. The other main body cavity is the ventral body cavity. And as if you see on here, the ventral body cavity is divided into a space located inside the chest. We call all this space, it's the thoracic cavity. If you remember, related to the chest is called thoracic. If I'm looking at the part of the ventral body cavity that is located within the belly, something related to the belly is called the abdominal. We're going to call this as the abdominal cavity. Down below in the pelvis, the ventral body cavity part that is located within the pelvis is what we call the pelvic cavity. It gets a little more complicated when we look at the thoracic cavity. The thoracic cavity is divided into three main cavities. Right and left pleural cavities. This is where the lungs are kept. The lung is surrounded by a serous membrane that has a double layer. Surrounding the lungs like this. This is called the pleura. And the pleura has an internal layer. This is called the visceral layer. And to the outside, this is going to be the parietal layer. In the middle, between the two lungs, we've got a space. We call the space is the mediastinum. Mediastinum. And inside the mediastinum, we've got a cavity where the heart is going to be located. We call this cavity is the pericardial cavity. Peri means around, and cardio means heart. So the cavity around the heart is my pericardial cavity. The part of the mediastinum that is located above the heart, so the heart is going to be located in here. The space located above the heart itself is included in the mediastinum with its cavity, its pericardial cavity. The part of the mediastinum that is located above the heart is what we call the superior mediastinum. So again, when we look at the thoracic cavity, it has two main cavities, the pleural cavity, the mediastinum. So we've got two pleural cavities for the two lungs, and in between the lungs, you've got the mediastinum. Inside the mediastinum, I have a special part for the heart. We call this space around the heart. This is a pericardial cavity. Above the heart, I have part of the mediastinum in which the blood vessels are traveling in and out of the heart. This is called my superior mediastinum. Another very important topic that we need to have an idea about here is the anatomical position. What is the anatomical position? So if I'm telling you which one is located superior, which one is located inferior, your head or your legs? Or your head or your neck? Which one is superior, which one is inferior? Head is superior, neck is inferior, right? But what if I am bending my neck like this? What if I'm bending my neck all the way down? what's gonna happen? So now the neck is above the head. So would I say it's superior to it? No. Why? Because simply we're gonna be always going back to the anatomical position. When we describe the relation between the different body parts, we describe them according to the anatomical position. What is the anatomical position? The person will be standing up, facing forwards. His palms are facing forwards. So this is the position according to which we're going to be describing the relation between the different body parts, not when I am lying down flat on the bed, or I am bending my neck, or I am flexing my spine. All right, so only this position is what we will be considering to describe the relationship between the different body parts. Caudal. All right, so when we look here at how do we describe the position of the different body parts in relation to one another in the anatomical position. In any shape has how many dimensions? What do you think? Here we're talking about not sides, we're talking about dimensions. So if I want to measure the volume of this cube, how many dimensions should I know? I need to know three dimensions. I need to know up and down, the height, sides, to determine the width, and the depth. Those are the three dimensions that I need to know, right? Anybody disagrees with me? All right, so with this in mind, how do I describe the relation between the different body parts? I need to use those three dimensions that exist. So I need to differentiate between what's up and what's down. What's going out, what's directed in. And we use, in order to tell whether a body part is directed outwards or inwards, the term medial and lateral. If I'm directed towards the midline of the body, it's gonna be medial. If I'm directed away from the midline of the body, it's gonna be lateral. But how about my limbs on here? If I wanted to describe the position of the hand in relation to the elbow, it's not lateral, it's not medial, it's not inferior and superior even. So we decided to create another relationship to describe the different body parts in the limbs, and this is by describing the position from the point of origin. So the point of origin of the limb on here, let's say for the upper limb, it's going to be the shoulder, and for the lower limb, it's going to be the hip joint. We're going to be describing the relationship according to whether I'm located closer to the point of attachment or the point of origin or away from the point of origin. So what would we call something closer to the point of origin? It's proximal. If something is located away from the point of origin, it's going to be distal. So again, we describe up and down as superior and inferior, cranial and caudal, related to the head, related to the tail. If I am describing something closer to the midline, it's medial. If I'm describing something away from the midline, it's going to be lateral. If I'm describing part of the limbs that is closer to the point of origin, it's proximal. If I'm describing a point in the limbs that is located away from the point of origin, it's going to be distal. So let me ask you a few questions on here. So the hip joint is blank compared to the knee joint. Hip joint is what compared to the knee joint? Remember, this hip joint is closer to the point of origin compared to the knee. So what do we describe this as? It's more proximal. How do you describe the heart compared to the lungs? So the heart is more blank compared to the lungs. What do you think? The heart is what compared to the lungs? It's closer to the midline. So what would we call this? The heart is closer to the midline than the lungs, right? Is it closer or is it away from the midline compared to the lungs? It's closer to the midline. So what would we call this? It's medial. How about if I'm comparing the lungs to the heart? So the lungs are what compared to the heart? They are located away from the midline. So they are more lateral. And of course, describing something forwards is anterior, backwards it's posterior. Other terms that we use for anterior and posterior. So anterior, so if I'm looking here at the person here. So this is going to be anterior. This is going to be anterior. And this is going to be posterior. Also, we call anterior is ventral. This is where I have the belly. And posterior is going to be related to the back. So we're going to call this as dorsal. So again, posterior for humans, it's going to be dorsal. For the anterior, for humans, it's going to be ventral. Why would I say in humans? Because if we're looking at, let's say, a cat, cranial for the cat, where the head is located, it's not superior, it's not up, it's anterior. And the tail for the cat, caudal, it's posterior. It's not inferior. Same concept here for the back. The back of the cat, it's not posterior. The back of the cat here, it's superior. So dorsal for the cat, it's going to be superior. And the belly here, it's not anterior. So ventral, it's not anterior. Like in humans, ventral for the cat here, it's going to be inferior. You see the difference? You see the difference? So ventral for humans is going to be ventral, it's going to be anterior. Dorsal is going to be posterior. No, it's not essentially for the limbs. It's not essentially for the limbs. By definition, proximal means closer to the point of origin, and distal is going to be away from the point of origin, not essentially for the limbs. All right. So for example, if I'm describing the small intestine, this is my stomach, this is the small intestine, like this. So where is the point of origin for the small intestine? It's where it gets connected to the stomach. All right, it's going to be variable according to where it's coming from. For example, here for the intestines, you're going to tell the point of origin because this is the first part that gets the electrical impulses for it to allow the peristalsis to take place. This is the first part that receives the food from the stomach, and so on. So we consider this part, it's the proximal part. So when I describe this portion, the C-shaped part of the small intestine, I would describe it as the proximal part of the intestine. See what I mean here? So it's going to be variable according to which organ. The easiest one to do, that's what I've used for the description here, is going to be the limbs. Because you can see the point of origin of the limb, it's the point of attachment to the rest of the body. If you're going away, it's distal. If you're going towards it, it's proximal. But this is, by definition, it's not only the limbs. By definition, it's whether it's closer or away from the point of origin. All right, so moving on to the cellular structure. The cell has three main components. Those are the plasma membrane. Any eukaryotic cell has three main components: plasma membrane, the nucleus, which will be the control center, of the cell, and all the remaining parts in here located between the plasma membrane and the nucleus is what we call the cytoplasm. So again, what are the three main components of any eukaryotic cell? You've got the plasma membrane, the nucleus, and the cytoplasm. The cytoplasm is going to be divided into three main parts. You've got the matrix, the fluid part. This is what we call the cytosol. You've got the embedded organelles within the cytoplasm, and you will have also some inclusions. The organelles are the machines that will be carrying on the cellular activities. Inclusions are just sitting in the cytoplasm. They are not going to be actively involved in mediating specific chemical reactions, like, for example, having pigments, having crystal salts within the cells. So those are considered inclusions. They are not organelles. If we're looking at the structure of the plasma membrane, we can see that the plasma membrane is formed of two main chemicals. Those are the lipids and proteins. We have three types of lipids. The first type of lipids on here has a phosphate head and fatty acid tails. This is called my phospholipids. Phospholipid. As you see, I have two layers of phospholipids. The head is a phosphate group, and the tail is a fatty acid attached to it. The heads are always directed towards the water, either the water on the outside of the cell, extracellular fluid, or the water inside of the cell, intracellular fluid. Compared to the fatty acids are directed towards the inside of your plasma membrane, away from the water. Why? Because the phosphate heads have charge on them, because those are the polar heads. Polar heads are hydrophilic. What I mean by being hydrophilic, philia is the opposite of phobia. Phobia is fear. Philia is love. So being charged, being polarized, having charge on me, the phosphate groups will love to get in contact with the water. They are hydrophilic. Compared to the fatty acids, like any lipids, if you put oil with water, can you mix them together? Would they love one another, or would they be separated always? What do you think? When you mix oil with water, would they be able to mix, or would they get separated? Always separate, right? Why? Because the lipids, like those fatty acids, are non-polar. And not having a charge will make me hate water or fear water. So we're gonna call those non-polar tails formed of fatty acids. Those are hydrophobic. Remember, phobia is fear. So I have hydrophilic polar heads and hydrophobic non-polar tails. The second type of fats that will be embedded in the plasma membrane, it's going to be these yellowish structures. Those are cholesterol molecules. The cholesterol molecules will maintain the fluidity and the flexibility of your plasma membrane. Your cellular plasma membranes are not stiff. So not with any minor trauma, you get cells rupturing. They are flexible, they are fluid due to the presence of embedded cholesterol molecules. The third type of fats that we can see here is going to be the fat to which we've got sugar units attached. And we call those ones are my glycolipids. What do I mean by glycolipids? Glyco means sugar and lipids, those are the fats. So we're looking here at sugar units attached to the phospholipids, so we call them glycolipids. So again, three types of fats. We got phospholipids, cholesterol, and glycolipids. We can see also two types of membrane proteins. A type of membrane proteins that will be traveling through the whole thickness of the plasma membrane. We call those proteins that are traveling through the whole thickness of the plasma membrane. Those are integral proteins. And some proteins might be loosely attached to the integral proteins. We call those are my peripheral proteins. Of course, you have been discussing the different functions of the proteins in your lecture course. All right, but here we just need to be able to identify those proteins when we see a diagram showing the plasma membrane. So again, we've got integral proteins which occupy the whole thickness of the plasma membrane, and peripheral proteins are loosely attached to them, either directed to the outside of the cell or to the inside of the cell. Moving
I have a
on
quick
to
question.
Mhm.
I mean, I wasn't here the last class. I'd email you about that. What would be the best way to take these notes? I mean, I'm looking at the PowerPoint right now on Canvas, but I'm not sure which would be the best way to take these notes and kind of.
So if you want any of my writings, you can just let me know at the end of class and I will give you a chance to take a screenshot of it.
Awesome. Okay.
All right. Also, you can find all the information that you need for the exams that have been posted in the need to knows lists.
Okay.
If you want those specific notes, just let me know at the end of the meeting, and I will just give you a chance to take a screenshot of them.
Okay. Thank you very much.
You're welcome. All right, so moving on to the second component of my cell, which is my nucleus. And if you see on here, the nucleus has a membrane. It's a double layered membrane. We call the membrane that will be surrounding the internal structures of the nucleus. This is my nuclear envelope. Looking here at the inside of the nucleus, we can see At the center of the nucleus, we have a dark spot. This is called my nucleolus. And the nucleolus, this dark spot in the nucleus, is going to be responsible for the production of the ribosomes.
I'm sorry, I have another quick question.
Mm-hmm.
So the quiz 1 diagrams, that's what's going to be on quiz 1?
Yes.
Okay. All right. Thank you.
You're welcome. All right, so nucleolus is going to be responsible for the production of an organelle that you're going to see in a second. This is going to be my ribosomes. Surrounding the nucleolus, you have a liquid form of the genetic material, and this liquid form of the genetic material is what we call the chromatin. So the genetic material, or DNA, is in the form of chromatin in most of the time inside the nucleus. If you can see on here, this nuclear membrane or envelope has pores, because those are the nuclear pores. For the information to be sent from your genetic material, from the chromatin, from the DNA, to travel to the outside of the nucleus. And if you've got external signals coming from outside of the nucleus, they can travel to the inside of the nucleus, passing through those nuclear pores to affect the activity of your DNA. If we're looking just attached to the nuclear membrane, we can see stacked membranous tubules. Those stacked membranous tubules, they have those reddish spots on top of them. What do we call those stacked membranous tubules surrounding the nucleus? This is what we call the rough endoplasmic reticulum. And those reddish spots on top of the rough endoplasmic reticulum is what we call the ribosomes. Those ones on here, those are my ribosomes. So the function of the ribosomes, it's to produce proteins. According to what would I be producing the proteins? According to the genetic code that is coming from your nucleus. So if you notice, you've got some ribosomes which are scattered around in the cytoplasm like this, and some other ribosomes are attached to the rough endoplasmic reticulum. So we call the ones attached to the rough endoplasmic reticulum, those are membrane-bound ribosomes. And the ones which are scattered freely in the cytoplasm, those are my free ribosomes. What is the difference in terms of function between the ribosomes, which are membrane bound, attached to the rough endoplasmic reticulum, and the free ribosomes? Both of them are going to be producing proteins. But the difference is, what type of proteins are you going to be producing? This is the main difference on here between the two. The free ribosomes are going to be producing soluble proteins that will remain inside the cell, compared to the free ribosomes are going to be compared to the membrane-bound ribosomes, they will be producing the proteins that will either be integrated within the plasma membrane, like what you see on here. The ones that will be integrated within the plasma membrane, those are produced by the rough endoplasmic reticulum ribosomes, or the ones that will be released to the outside of the cell, exported to the outside of the cell. So if we're looking again at those ribosomes, we've got two types of ribosomes, either free ribosomes or membrane-bound ribosomes that are attached to the rough endoplasmic reticulum. What happens to this protein after it gets produced from the ribosomes attached to the rough endoplasmic reticulum? It's time now for me to trim and condense those proteins, and for me to send them to their destinations. The organelle that is going to be responsible for the condensation, the trimming of the protein, and the packaging of those proteins that we produce by the ribosomes in the rough endoplasmic reticulum, this is what we call the Golgi apparatus. The Golgi apparatus importance is gonna be, it is the one that will be determining the fate of the protein. So for example, if I am producing a protein that will act as a digestive enzyme for the bacteria, would you like to keep it stored in a vesicle until you get a bacterial infection for you to release this digestive enzyme, or would you like to keep releasing it to the outside as it gets formed? What do you think? Would you like to keep it stored in a vesicle until you need it, or would you like to keep releasing it to the outside? Keep it stored for a while until you need it in a vesicle. What would you like to do if you're producing a protein that will be acting as a channel for your plasma membrane? Would you like to store it inside your cell in a vesicle? Would you like to get it integrated within the plasma membrane, or would you like to release it out from the cell? What do you think? If you are producing a channel, a protein that will form a channel for ions to travel inside your cell, would you like to store it inside a vesicle? So I have the channels like this, stored in a vesicle inside the cytoplasm. Or would you like me to integrate it within my plasma membrane for it to act? Or would you like me to release it to the outside in the blood, let's say? So this channel won't have any function. If it remained in a vesicle inside the cell, it won't function. If it got released outside of the cell, it won't function, because it's a channel that allows ions to travel through the plasma membrane. So those protein channels need to be integrated within your plasma membrane. So who's going to be determining the fate here of such proteins? It's going to be the packaging that they get from the Golgi apparatus. So some packaging of a protein will allow it to become integrated within the plasma membrane like a channel. Some packagings that I will receive will fuse with the plasma membrane to allow you to release the contents, to allow this protein to get released to the outside of the cell. Some packaging will keep the protein stored inside a vesicle. So again, what is the importance here of the Golgi apparatus? It's going to be condensing, trimming the protein, and package the protein. And why is the packaging of the protein is so important? Because simply, it's the one that's going to be determining the fate of this protein, whether it's going to be integrated within the plasma membrane, stored in a vesicle or released to the outside of the cell. Think of it this way. So where would you put a diamond ring? In a jewelry box, or would you put it in a plastic bag from Walmart? What's going to happen to this diamond ring if you put it in a plastic bag from Walmart? Would it be preserved, or will it be lost in the trash? Yeah, you have a much greater chance to lose it if you just put it in a plastic bag from Walmart. All right, so the packaging here of the ring did determine its fate. All right, the same concept on here. The packaging that I'm giving to the protein is what's going to be determining the destination and the fate of this protein. Like what vesicles do I keep inside my cell? What proteins do I keep inside the cell? Like, for example, if I am storing digestive enzymes in a vesicle, we're going to call those as lysosomes. What are the lysosomes? Those are membranous organelles in the form of vesicles containing hydrolytic enzymes. What are the hydrolytic enzymes? They are digestive enzymes. So what do you need to break down using those lysosomes? If you need calcium from your bones, for example, you go and break down the bone to release calcium. If the cell is not functioning, it's going to start to destroy itself using those hydrolytic enzymes inside the lysosomes. If, for example, I did engulf a bacteria and I want to destroy the bacteria that I did engulf, what will I do? I will digest it with the lysosomes. All right, so again, what is the function here of the lysosomes? It contains hydrolytic enzymes that will help you to digest different things according to the state of the cell. Another vesicle that stores other types of proteins. This time, those proteins are acting as enzymes responsible to allow oxidation and reduction of superoxide molecules. What are superoxide molecules? Those are like the hydrogen peroxides that you see on here. So water is neutral. Water is neutral. It's H2O. But H2O2, this is hydrogen peroxide. Have you ever seen hydrogen peroxide before? Yeah, where can we find it? What do you think? Yeah, drugstore. And what do we use it for? To clean wounds, right? It's a toxic substance. In the process to produce energy, we need to consume oxygen to break down the nutrients. And when we consume this oxygen should be binding to hydrogen and becomes water. But sometimes I will be forming those highly active chemicals instead of producing just water, because those are superoxide. But those superoxide, when they accumulate within the cells, what's going to happen to the cells? The cell is going to be destroyed because those are relatively toxic. So in order for me to make them more chemically stable, I need enzymes to act on them, and those enzymes are going to be stored for me in vesicles. We call those vesicles containing the oxidative enzymes and the reductases. Those are my peroxisomes. Peroxisomes. So again, what we've seen so far, we've seen the nucleus with the nuclear membrane, and at the center of the nucleus, we've got the nucleolus, which is going to be responsible for the formation of the ribosomes. Attached to the nuclear membrane, we have stacked membranous tubules, on top of which we've got ribosomes. Some other ribosomes will be scattered around in the cytoplasm. We call the ones scattered in the cytoplasm, those are the free ribosomes. The ones that are attached to the outside of the rough endoplasmic reticulum, those are the membrane-bound ribosomes. Both types of ribosomes are going to be needed for the production of the proteins. The only difference between the two is that the free ribosomes are going to be producing proteins which are soluble and they will be kept inside the cell, compared to the ones that are attached to the rough endoplasmic reticulum. Those are going to be responsible for the formation of the proteins that will either be integrated within the cell membrane or that will get exported outside of the cell. Two examples on here of proteins that will be stored in vesicles inside the cell, like the lysosomes. Lysosomes contain hydrolytic enzymes, like the peroxisomes, which contain the oxidative enzymes, which are necessary to get rid of the superoxide molecules that are resulting from the metabolic reactions. Another organelle formed of stacked tubules. This time they won't have any ribosomes on top of them. That's why we're gonna call those, this is gonna be my smooth endoplasmic reticulum. Smooth endoplasmic reticulum function is gonna be variable according to where it is located. So in, for example, the liver cells, it's gonna be responsible to metabolize the fat. It's going to be responsible to detoxify the toxins. In the muscle cells, another example, is going to be responsible to store the calcium and release it for the muscle to contract. Looking at another type of organelles here, it's going to be a double membrane organelle, a double membrane organelle. You see an outer smooth membrane and an inner convoluted membrane. We call this organelle, it's the mitochondrion. Mitochondria are going to be responsible for the production of cellular energy. This is where the combustion of the nutrients going to be taking place, and this is where most of the cellular energy going to be generated. We also have a skeleton that is going to be formed by different types of protein fibers, like the microtubules and the intermediate filaments, which also going to be playing an important role in the transport of the different organelles, and the movement of the different organelles within the cells. All right, so let me give you one last quick review of this diagram showing the cell. Again, we mentioned the plasma membrane. We stated the nucleus with the nuclear envelope. At the center of the nucleus, we've got the nucleolus, which is going to be responsible for the formation of the ribosomes. Filling the nucleus, we've got the chromatin, which is the liquid form of the genetic material, your DNA. We've got nuclear pores through which the codes that you want to transform into a protein going to be traveling from the nucleus to the rough endoplasmic reticulum around it. Rough endoplasmic reticulum, this is an organelle that is formed of stacked tubules, on top of which you've got ribosomes. Ribosomes exist either free or membrane bound. They are responsible for the protein production. Proteins that are produced from the rough endoplasmic reticulum, those are going to be either integrated within the cell membrane or exported to the outside of the cell. Those proteins will be trimmed, condensed, and packaged in the Golgi apparatus. Remember, the packaging will be determining the fate of the produced protein. If we're looking at two examples of vesicles that will contain proteins and will remain inside the cell, we've got the lysosomes, which carry hydrolytic enzymes, and we've got the peroxisomes, which are going to be carrying oxidative enzymes that will allow you to get rid of the superoxide. Another organelle formed of stacked tubules, but without ribosomes on top of it. This is going to be the smooth endoplasmic reticulum. And again, the function of the smooth endoplasmic reticulum going to vary according to what organ cell is it located in. In the liver, intestinal cells, it's going to be responsible for the metabolism of the fat. In the liver cells is going to be, and kidney is going to be responsible for the detoxification of the toxins. In the muscle cells, it's going to be responsible to store the calcium and get it released at the time of contraction. We're looking next to another organelle, which is the mitochondria. The mitochondria are going to be responsible for the production of the cellular energy, again, resulting from the combustion of the nutrients in the presence of oxygen. The cellular energy is going to be in the form of adenosine triphosphate or ATP. Any questions? Any questions? Anybody still here?
Yes, of course.
All right, so we can have a 5 minutes break. We can have a 5 minutes break. We come back, discuss the cell cycle. All right, any questions before we leave for a break? Questions? All right, so we'll see you in 5. All right, welcome back. You just want to double check if you still can hear me and if you still can see the PowerPoint presentation on the screen. All right. So moving on to the cell cycle. What is the cell cycle? Cell cycle is the life cycle of a cell. The cell cycle is divided into two main phases, the interphase and the mitotic phase, or the M phase. So interphase is the time that the cell used to grow, and it's going to be divided into three main subphases, G1 or growth 1, S phase or growth and DNA synthesis phase, and G2, which is the second growth phase. In G1 and G2, what the cell is actually doing, it's increasing the number of organelles, it's increasing the amount of cytoplasm, preparing for the cell division. What's special about the S phase of the interphase is this is the time when the genetic material, the DNA, is going to be replicated. This is when I have the replication of my DNA. So if a cell is going to be dividing into two daughter cells that resemble the mother cell in all aspects. So this means what for me? This means that the two daughter cells need to contain the same genetic material, the same DNA as the mother cell. So in order for me to divide into two daughter cells that will contain all of the genetic materials that I have, what I need to do, I need to make a copy of my genetic material in order to give each of the two daughter cells a copy of this genetic code. This is what we call the DNA replication. And again, the DNA replication is not taking place like what some of you might think during the division part. It's taking place during the growth part. To be more specific, it's going to be taking place during the S phase of the interphase. After I grow, after I duplicate my genetic material, what will I do next? I will start the cellular division. What if the cell does not divide? It's a permanent cell. Like what permanent cells that we've got? Like the nerve cells, for example. You have a certain number of nerve cells, and that's it. They can't regenerate, they can't replicate. Like the muscle cells. You again have a limited number of muscle cells that can't replicate, and this includes the skeletal muscle cells and the cardiac muscle cells. Smooth muscle cells can replicate in certain conditions. So those cells gonna remain in a G0 phase. They never get to a mitotic phase. They never divide. Those again are the permanent cells, like the nerve cells, the neurons, and the muscle cells, like the skeletal muscle cells and the cardiac muscle cells. how the cell looks like when it's growing during G1, S, and G2, or the interphase. This is how the cell looks like. The cell has a plasma membrane, has a nuclear membrane around the genetic material, and the genetic material is going to be kept in the form of chromatin, this liquid form of the DNA. During this phase, while I'm preparing to divide the cell, I have something that we call the centrosomes. Each of the centrosomes has two centrioles. Those are formed by microtubules, which are protein tubules that will be responsible for the formation of the fibers that will be pulling the genetic material to the two opposing sides of the cell. So again, what we're looking at in here, we're looking at the cell structure during the interphase, the growth phase. So during interphase, I have a nuclear membrane. I have genetic material in the form of chromatin. And the nucleolus, which as we mentioned earlier, was responsible for the production of the ribosomes, gonna be inside the nucleus. When I start this cell division, what's gonna happen? I start first with an early phase of the mitosis called prophase. During prophase, the two centrosomes gonna be migrating to the two opposing poles of the cell. In the same time, the chromatin, which is the liquid form of the genetic material, gonna be condensed into chromosomes. Each chromosome has two arms. We call those two arms are the sister chromatids. And the two sister chromatids are attached to one another by the centromere. So again, what happens during early prophase, the first phase of the cell division or the mitotic division? During early prophase, you've got the centrosomes migrating to the two opposing poles of the cell. The nucleolus disappears. The genetic material in the form of chromatin gets condensed into chromosomes. Each chromosome has two sister chromatids, which are attached by a centromere. Any problems so far? Any questions so far? All right, so late prophase, what happens? The nuclear membrane degenerates, and those centrosomes will be forming mitotic spindle fibers. Those fibers on here are gonna be traveling in the cell and will bind those chromosomes at the centromere. This is again during late prophase. Those mitotic spindle fibers gonna start to pull the chromosomes that they are attached to. And as I keep pulling the chromosomes to the two opposing poles of the cell, the chromosomes gonna be arranged at the equator of the cell. This line that gets formed by the centromeres of the chromosomes is what we call the metaphase plate. The line that gets formed by the centromeres of those chromosomes is what we call the metaphase plate. And we call this phase in which I'm arranging the chromosomes at the equator of the cell, it's my metaphase. As the mitotic spindle fibers keep pulling the two sister chromatids away from one another, the centromeres that were attaching them together will rupture, and the two sister chromatids gonna be migrating to the two opposing poles of the cell. We call this phase in which I have rupture of the centromere and migration of the two sister chromatids away from one another, it's the anaphase. Now I've formed two groups of chromosomes on each side of the cell. What happens next? Those mitotic spindle fibers that were attaching the chromosomes now will degenerate. The nucleolus appears next to those chromosomes, and a nuclear membrane starts to get formed around the two groups. So now the cell has two nuclei. We call this phase is my telophase. During late anaphase and during telophase and after, I will start to form a constriction ring at the equator of the cell for me to split the cell into two separated cells. We call this phase in which I am forming a constriction ring that splits the cell into two. This is my cytokinesis. We mean by cytokinesis is the cell division. Mitosis is the nuclear division, the division of the nucleus. So again, what happened? In mitosis, I started with prophase. During prophase, what happened? The chromatin gets condensed into chromosomes. The centrosomes are going to be traveling to the two opposing poles of the cell, forming their mitotic spindle fibers. The nucleolus disappears. The nuclear membrane starts to degenerate, and the mitotic spindle fibers are going to bind to the centromeres of the chromosomes. Centromeres are attaching the two sister chromatids of the chromosome to one another. Each of those arms is going to have a similar copy of the genetic material like the other. So those are simply two copies of the chromosome attached to one another. What happens next? The mitotic spindle fibers gonna start to pull the chromosomes to the two opposing poles of the cell. This will cause the arrangement of the chromosomes at the equator of the cell. As I keep pulling those chromosomes away from one another, the centromere will be broken down, and the sister chromatids gonna be traveling to the two opposing sides. We call this phase of the mitosis, this is the anaphase. Now I've formed two groups of chromosomes. The chromosomes will return back to the chromatin appearance. The nucleolus will reappear. The nuclear membrane will be formed, and the mitotic spindle will degenerate, forming two separate nuclei. We call this phase again, it's my telophase. During late anaphase and during telophase and after, you're gonna form this contractile ring that will be splitting the cell into two separate cells. So we always want one of each chromosome when splitting. Yes, exactly. So for the human cells, we've got 46 chromosomes. So when your skin cell, for example, is regenerating, replicating, it's gonna be forming. So let's imagine you've got 46 chromosomes like this. Before the cell division, what will I do? I will be duplicating my genetic material. So now I still have 46 chromosomes, but I have two copies of each of those 46 chromosomes. All right. So during the cell division, what's going to happen? The centrosome is going to be putting this arm toward one side and the other arm towards the other side. So now when they separate, they are separated, we're going to call each one of them not chromatid anymore. Each one of them is gonna be called a chromosome. So how this is a chromosome and this is a chromosome. Although this one has two arms, this one has a single arm. Simply, a chromosome is the set of information. If I have two copies of the same set of information, I still have one book. If I have two copies of a single book, what is the amount of information that I have? It's one book. So it's one chromosome on here. I have only one book of information. Even if I have 1,000 copies of it, it's still, in terms of information, one book. All right. So if I've given those two copies of the same book to two of my sons, so each of them gonna have the same books that I've had. The only difference is that I did have two copies of it. So we call a chromosome not according to the amount of copies, but according to the set of information included. So here I have a complete set of information. Here I have a complete set of information. Here I have a complete set of information. All right. Does this make sense? Does this make sense? All right. Any questions? Any questions? So again, let's go to the cell cycle. We started with G1. I'm growing, increasing the cytoplasm, increasing the organelles. I'm duplicating the genetic material. So this is a time where I will be forming the other arm, but I can't see it because if you remember, during interphase, it's all liquid in the form of chromatin. When it's condensed at the beginning of the mitosis, during early prophase, when it's condensed, now I can see the two arms of the chromosome. Each one has a copy of the genetic material on this specific chromosome. So after growing and duplicating the genetic material, the cell divides, passes first through prophase, then metaphase. The chromosomes are arranged at the equator of the cell, and we call this line that gets formed, again, it's my metaphase plate. Once the centromeres rupture and the two sister chromatids get separated from one another, we call this is my anaphase. You form two sets of chromosomes, you reform the nuclear membrane around each set, the nucleolus reappears. This is the telophase. Remember, cytokinesis is the cell division. This is the formation of the constriction ring in the middle of the cell. Cytokinesis starts during late anaphase, during telophase, and after telophase. Now I've got two cells that look like the mother cell, two copies of the cell. Questions?
You said cytokinesis is it starts after what?
What exactly starts after what?
I was writing down what you.
I can't hear. So can you unmute?
Oh, I'm sorry. Yeah, I thought I was muted. So I just didn't understand what you were saying about cytokinesis. Is basically what I was asking.
So cytokinesis, if you see on here, this constriction ring that we've seen down here, that would be dividing the cell into two cells. All right, this process is what we call the cytokinesis. This cell division does not start after telophase. After you complete the telophase, the formation of the nuclear membrane and the reappearance of the nucleus, you don't have to wait until all this is completed to start the separation of the two sides of the cell.
Okay, that's exactly what I was wondering. Thank you.
All right, so actually it starts, you see on here on this graph or on this diagram, you see cytokinesis starts during late anaphase. It
Awesome.
keeps going during telophase, and it is completed after telophase. So I've lost connection on here, just 1 second, please.
I don't know if this would help you guys, but I had a teacher kind of break it down to me in a weird way a year ago. And she was like, think of cytokinesis as basically like the labor, like you are birthing a new cell. So it's like it's separating, it's a new one is coming. Like that's like the end of it. So it's like you think of cytokinesis because when women are in labor, they can come off psycho because they're in pain, like, you know. So she kind of broke it down in that way, and I don't know if it would help you to look at it that way. So.
All right,
Thank you,
for
Avery.
sharing this.
Thank you.
You're welcome.
All right, so you see on here, starts during late anaphase, keeps going during telophase, and it's done after, it's completed after telophase. All right.
All right, thank you.
You're welcome. All right, so let's move on to our next topic, which is the tissues. All right, so just 1 second on here. So if you remember from the introduction of this course, from your lecture course, what was the most basic building unit of your organs? Atoms, right? Those are the smallest building blocks of the different chemicals. These atoms are different from one element to another. So like the carbon, nitrogen, hydrogen, oxygen, iron, copper. So you have atoms. And those atoms most likely do not exist in nature by themselves, free atoms, because they are not chemically stable. So in order for them to be chemically stable, they will need to form chemical bonds. And the formation of those chemical bonds will allow them to form molecules, which are more complex chemical structures. Like for example, if I did attach 6 carbons to 12 hydrogens to 6 oxygen, I will form a large molecule here. We call it glucose. Similarly, we're going to be attaching the different molecules to one another to build fats, to build proteins, carbs, and nucleic acids. All those will form together or will share in the formation of your organelles. Like what organelles that you know of, like the ribosomes, the rough endoplasmic reticulum, the plasma membrane, the nuclear membrane, DNA. So everything in your cells is going to be built by those various chemical molecules, which are built by the atoms. Those organelles will share together in building the smallest living unit of your body, which is the cell. The smallest living unit here is a cell. But the most basic chemical structure is the atom. Cells of the same type will share together and will form a tissue. Different types of tissues will form together an organ. And organs, if you remember, that are going to be responsible to produce a specific function will work together, forming an organ system, like the cardiovascular system, like the respiratory system, like the integumentary system, nervous system, skeletal system, muscular system, and so on. So we've got different organ systems that, when working together, will build up an organism, like a human being, for example. So if we're looking at in here, looking today at the tissues. So if I put the tissues in context, they are built by similar cells. And those tissues, multiple types of tissues, different types of tissues, will be forming together an organ. So when I look at the tissue under the microscope, because I can't see tissues with my naked eye, so I need a magnification power to increase the size for me to be able to describe the structure. So when I look at those tissues will not exist on their own. So you won't see just one type of tissue because you're cutting parts of the organ that are formed of different types of tissues. So when we look at a tissue, under the microscope, it won't exist by itself, 100% by itself, because we're cutting through an organ that is formed by different types of tissues. The building units, again, of the tissue, it's going to be the cells. How many types of tissues do we have in our organs? We've got 4 main types of tissues. All your organs are built by those 4 main types of tissues. First type of tissues that we're looking at, and we're going to discuss today, it's the epithelium, or the epithelial tissue. Second type of tissues, it's called the connective tissue. Third type is the muscle tissue. And lastly, it's going to be the nervous tissue. Each one of those tissues is going to be characterized by specific characteristics and will be responsible to produce specific functions. So if we're looking at the epithelium, it's going to be either a glandular epithelium, what I mean by glandular epithelium, the cells of this epithelium going to share together to form a secretory organ, and we call those secretory organs that would be formed by the epithelium are glands. So again, epithelial tissue can form glands, and the epithelium that form glands, we call it glandular epithelium, and we're not concerned with it in our lab. So we will not be differentiating between the types of glands, what are the shapes of the various glands, so we're not concerned with this part in our lab. The second type of epithelium is the epithelium that will be responsible for the formation of the coverings and linings of the different organs. Surface epithelium. So any organ is covered from outside by epithelium, and if it has a cavity in it, this cavity is lined with epithelium. So again, when we look at epithelium, how many possible types of epithelium can we have? We have either glandular epithelium or the epithelium responsible for the formation of the coverings and linings of the different organs. The connective tissue, what do you think is it going to be responsible for? In a very simplified way, from its name, what do you think is going to be the primary function of the connective tissue? From its name, what do you think is a primary function? Connecting the other tissues to one another. Exactly. So the main function, we're going to have many functions of the different types of connective tissue, but the general function of the connective tissue is that it attaches, it connects the other three types to one another. Because remember, an organ is not simply formed of one type of tissues, it's going to be formed of different types of tissues that will share together. So in order for me to connect the epithelium to the muscle, I will need connective tissue in the middle. For me to connect epithelium to nervous tissue, I need connective tissue in the middle. I need to connect, attach the various tissues to one another for me to be able to build the organs. Again, this is just a general function, but we're going to get to the details of the various types of connective tissue in next class. Muscle tissue, what do you think is the main function and relying on the main characteristic of the muscle cells? What's so special about the muscles compared to any other cell? Movement. What else do you think?
This might be stupid, but they can grow and expand.
We do have other cells like fat cells, they can still grow, but what allows the movement to take place? What do you think? What do you think? What allows the movement to take place is their ability to contract. What is so special about the muscle cells? Cardiac muscle cells, skeletal muscle cells, smooth muscle cells. What's common in all those muscle cells is that they are able to contract. And what do I mean by contraction? If I have a bone in here, so let's say this is my humerus, like this. This is my radius. This is the ulna. And this is going to be the scapula like this. All right, so if I told you you've got a muscle that gets attached like this in the scapula and travels down to be inserted in my radius. Like this. What do we call this muscle? All of you will know it. This muscle with two heads located in the anterior aspect of the arm. The most famous one, yeah, biceps. This is my biceps, right? So how the biceps is performing its action. What is the action of the biceps? So when you go train your biceps, it's my biceps brachii. What I'm describing here is the biceps brachii. So what is the function of the biceps? What action will it be performing? What action will it be performing? It's going to be pulling the forearm like this. So what happened in the muscle? If I am looking at the whole muscle, this is my biceps. It's attached to one point to the radius, the other point to my scapula. When I contract, what happens? I am shortening the length of the muscle. This is what is muscle contraction. Muscle contraction is the shortening in the length of the muscle. So what happens as a result? This point that I'm attached to is going to be pulled towards the other point. So this one is fixed. It can't move. This one is mobile, so it can move. So what's going to happen? This point is going to be dragged towards the other point, and the movement takes place, allowing flexion. All right, so again, what is a muscle contraction? What is so special about the muscle cells? What's so special about the muscle cells is that they are able to perform a contraction. And what's a muscle contraction again? Can you remind me? I forgot. What's a muscle contraction? It's shortening in the length of the muscle. All right, so this is my third type of tissues. And lastly, the last type of tissues that we're going to be discussing in this lab, it's going to be the nervous tissue. What is the most important characteristic, do you think, of the nervous tissue? Exactly. I have cells that got the ability to send out messages in the form of electrical impulses. So again, what are the four main types of tissues that we've got building up all the organs in the body? Electrical charge, and the variation of the charge is what we call an impulse. All right, the variation of the charge is what we call the impulse. Alicia, we're gonna study this in details later on in the nervous system. All right, so again, four types of tissues: epithelium, connective, muscle, and nerve. So the ones that we're concerned with in the epithelium is the ones that's going to be responsible for the formation of the coverings and linings. So what are the characteristics of the epithelium that is going to be forming the coverings and linings? So forming the covering of an organ or lining the inside of a cavity. This tells me that those cells forming the epithelium are going to be exposed to irritation because they are forming the outer barrier of the organ, right? Am I exposed to more or less irritation compared to internal tissues? If I'm forming the outer barrier, I'm exposed to more irritation, right? So more irritation, what's going to happen? If I did have blood vessels within the epithelium, if the epithelium did have blood vessels here, what might happen? Vessels would be damaged and I would start to bleed. But this doesn't happen. Why? Because simply the epithelium, very first important characteristic of the epithelium is that it's avascular. It doesn't have blood vessels. So how does it get its nutrients and oxygen? Would those cells be able to survive if they are avascular, if they don't have blood vessels for themselves? Yes, they are able to survive because they are supported by the underlying connective tissue. So you've got connective tissue below any epithelium, and through this connective tissue, fluid carrying oxygen and nutrients gonna be diffusing up towards your epithelium. It's diffusing through the epithelium. Intercellular connections will allow this fluid to travel, yes.
Are they flowing through like the same way as like a channel, like when you're dealing with the membrane? Is it like it
Not
has, or just it's loose enough where it can just go right through?
so it's all gonna be depending on the hydrostatic pressure more than the osmotic pressure. So what you're talking about is called the osmotic pressure. So the fluids will be moving by the effect of the difference between the concentration of a solid on one side compared to the other. So the fluid is going to be moving from one side to the other according to the concentration of solids.
That's right. All right, thank you.
All right, but here it's a pushing force. The heart contracts, creating blood pressure, and this blood pressure is going to be pushing the fluids to travel from the vessels here within the connective tissue. The connective tissue is vascularized, it has blood vessels, and it pushes those fluids through the connective tissue, and the fluid will be able to diffuse to the cells of the epithelium.
Thank
All right,
you.
you're welcome. All right, so looking here at another characteristic of the epithelium is that the epithelium, as we've mentioned here, is forming the coverings and linings of the different organs. So it has two sides. One side of the tissue is going to be attached to a membrane. We call this membrane, it's my basement membrane. And we call this side that's going to be connected to the underlying tissues that I'm covering. This is my basal side. And the other side that is not attached to anything, I have an empty space here, either the outside of the organ or the lumen of the organ. I have just space on the other side. This is going to be the top of the tissue, and we're going to call this one, it's my apical side. So we call this characteristic of the epithelium that the epithelium is polarized or having polarity. What I mean by having polarity, I have two sides. I have apical side and I have a basal side. How would I differentiate? The apical side is not attached to anything. It's exposed to the lumen of an organ or to the outside of an organ. The basal side of the tissue, it's going to be attached to the underlying tissues. This is another very important characteristic. One more important characteristic that we need to remember related to the epithelium, as I get exposed to a lot of irritation, I don't have blood vessels to not bleed. How about my ability to replace the injured cells? What do you think? Do I have high proliferation or proliferation power? My ability to replicate my cells, to duplicate my cells. I will need to have a high proliferation power. I am able to rapidly divide my cells. So you can see it in your skin. So it doesn't take time for you to cover an injured part in your skin. Cells are rapidly proliferating, replicating, and they will cover the injured site within days. All right, so how can I differentiate between an epithelium and the other tissues? If I would recommend just one thing to find to be able to differentiate between the epithelium and the rest of the tissues, is to look for a space that is lined with cells. If you find the space lined with cells, it's an epithelium. What do you think? Here. You have a space lined with cells. What do you think? Space lined with cells. Just tell me yes or no. Do you see a space that is lined with cells? Space lined with cells. Well, this one. Space lined with cells. You see space lined with cells. Can you see an empty space here? And it's lined with cells. How would I know that those are cells? First thing that tells you that those are cells, they have boundaries. You can't see the plasma membrane. Another thing, if you can't see the boundary of the cell, is the presence of the nucleus. All right, inside the cell, you've got the nuclei on here. So this tells me that I'm looking at cells. Let's look at this one on here. What do you think? Do you see a space lined with cells? No. Can this be an epithelium? No. How about this one? Do you see a space lined with cells? The lining of the space is formed by cells. It's not epithelium. Well, this one. Space lined with cells. What do you think? Yes. Where is it? Can you annotate? Can you mark down from your side? This one on here? This is what you're pointing to? So this is a space, all right? But where is it lined with cells? Where are the cells lining the space? You see cells that are surrounding the space, lining the space? What do you think? No. Space here, but where are the cells lining the space? I have space here, kind of. It's actually filled with tissue. Right, so I don't have epithelium here. No epithelium that I'm looking at. How about, let's see on here. How about this one? Space, yes, I do have a space. Is it lined with cells? You have cells scattered around. They are not lining the space. How about this one? What do you think? Do you see a space lined with cells? Space lined with cells. Space lined with cells. What do you think? Space lined with cells. Yes or no? Is this an epithelial tissue? No, why? Although some people actually get confused with this. This is a space, yes, this is an empty space, but actually it's not lined with cells. It's inside just one cell. See, one nucleus. This is the compressed nucleus of this cell. So this is an empty space that was not empty. It was filled with fat at a certain point, but during the preparation of the tissue, the fat is melting out of the cell and it became like an empty space. So yes, this is a space, but not lined with cells. It's inside the cell. Let's have a look on here. What do you think? Do I see cell space lined with cells here? No. Well, this one. No. I see fibers. So it's not epithelium. It's not epithelium. This is the main thing I want you to be able to figure out at this point. So what do you think about this one again? Is this epithelium, yes or no? Yes. About this one. Do I see a space? And this space is lined with cells. Again, how do I know that those are cells? Boundaries and nuclei. So do you see a space? This space is lined with cells. It's epithelium. Well, this one here. Space lined with cells. Space, cells, lining the space. All right, so how can I differentiate, or how do I classify the epithelium? How do I classify the different types of epithelium? The first factor I use to classify the different types of epithelium is by checking the number of cell layers. So these cells are arranged in layers on top of one another. So if I do have just one layer of cells, just one row of cells forming the epithelium, it's going to be classified as simple. If I see more than one layer of cells on top of one another, lining this space that I'm looking at, more than one cell layer, it's going to be classified as a stratified epithelium. So this is the first characteristic according to which we're going to be classifying the epithelium again, according to number of cell layers. If you've got one cell layer, it's simple. You've got more than one cell layer, it's stratified. How about one more characteristic is the shape of apical cells. What this means? Apical cells are the cells forming the free border, forming the outside. They are not attached to anything on this side. Those are my apical cells. So what determines the type of epithelium? Second factor that I'm using here is the shape of the apical cells, the cells at the top. Those cells might be either flat cells, so we call those are, this tissue is going to be classified as squamous. Those apical cells might be cube-shaped cells, and how would I tell that those are cube-shaped? The height is almost equal to the width of the cell. So we classify this epithelium, which cells are cube-shaped. This is a cuboidal epithelium. Cuboidal epithelium. So you've got either squamous, cuboidal, or if I do have tall cells, so the height is much greater than the width of the cell. This epithelium is going to be classified as a columnar epithelium. So again, what are the two factors that we use to determine the type of the epithelium forming the coverings and lining? The first factor is according to the number of cell layers. If I do have one cell layer, it's simple. If I have more than one cell layer, it's gonna be stratified. Shape of the apical cells. If the cells are at the top of the epithelium are flat, the epithelium is going to be classified as squamous. If the cells at the top are cube-shaped, it's going to be cuboidal. If they are tall, it's going to be columnar. So can you tell me the type of the epithelium that we're looking at in here? What do you think? Without having any previous information, according to just what we've mentioned. So how do we classify? Two factors, two things that we use: number of cell layers and the shape of the cells, shape of the apical cells. So first, do I have a space lined with cells? Yes or no? Yes, so it's epithelium. How many cell layers are forming the lining? So this is the whole space. How many layers are forming the lining? It's just one layer. How more than one? So some people would see this is one layer, this is another layer. If you do have a space between the layers, it's not more than one layer. All right, you have only one layer lining the space from inside. See, this is just one cell. Stephanie, does this make sense? So I have no other cell on top of me. I have this space. All right, so what do we classify the epithelium that is formed of one cell layer? Can you remind me? Simple. Exactly. And what do you think is the shape of those cells? Are those cells flat, cube-shaped, or tall cells? Squamous, cuboidal, or columnar? Flat. So I am formed of one row of flat cells on here. So this is going to be a simple squamous epithelium. So on the exam, you're going to have those or similar diagrams provided by the college. And you're going to be, I will be asking you, identify the type of epithelium or identify the type of tissue. So what's going to be the type of tissues that we're looking at? It's a simple squamous epithelium. Easy, easy. Easy, yes, no. I have no idea what you're talking about. Yes. All right, so let's have a look on here. What do you think? First thing you need to ask yourself is what Do I see space lined with cells? Yes or no? Space lined with cells. Yes, so it's epithelium. It's epithelium. So let's look at this epithelium. So this is my space, and those are the cells lining my space. On here, I can't see the boundaries, but I can see nuclei. So those are the cells. All right, so how many cell layers are lining the space for me to be able to tell whether it's a simple or stratified epithelium? How many cell layers? So we're looking at a tube, and this tube is lined with one layer of cells. You can't say that this here's a cell, this is a cell, and this is a cell, another cell on top of it. You do have the space in the middle. Those are not on top of one another. So it's actually a formula of how many cell layers? You've got one cell layer. So it's going to be simple. And what do you think is the shape of the cells? So let's have a look at another cell where I can see clear borders. What do you think? This is the height of the cell, and this is the width of the cell. Is this flat, cube-shaped, or tall cell? Is it squamous, cuboidal, or columnar? It's more like a cube, so it's going to be simple cuboidal epithelium.
So I'm a little confused because the last time we looked at the cells that were in the layer. So now we're looking at the cells that are inside of it to determine, because if I was looking at the layer, I would think squamous because of the thin outside layer. What made you look at the cell inside to determine that?
So you mean you want to look at those cells?
Okay. I'm
Do
seeing
you
it now.
want
All right.
to
All right.
look at
Thank you.
those cells here?
Okay.
Or what?
Because, well, the one that you have marked up with your blue marker, earlier you said that that wouldn't count as a layer. So in my mind, I would be looking at the outside blue line that you're circling around the whole cell. And I would look at those for the shape, right?
Yes. And this is a similar one, but I have here multiple tubes next to one another, lined with the same type of epithelium. So all of them have the same type of epithelial lining. Why I've chosen this one to identify the shape, because this one, if I'm removing here, it has clear borders.
Okay.
But this
All
one,
right,
when I
thank you.
remove here the markings, it doesn't show clear borders. That's why I've chosen this one to check the shape of the cell. But if I've checked this one from the beginning, it's going to be the same. All
Okay,
right?
thank you.
I can check this one, no problem with that. All right? But I started with this one because it has a larger space for me to tell you that I do have a large space that you can see. And then I moved to this one here to show you the shape of the cell, because this one has a clear border. What if I couldn't find a clear border? How would I tell the shape? It's according also to the shape of the nucleus. So if you don't rely on this unless you can't see the borders of the cell. You can guess the shape of the cell by looking at the shape of the nucleus. If the nucleus is flattened like this, so most likely the cell is going to be flat. If the nucleus is circular like this, like what you see on here in those cells, most likely the cell is going to be cuboidal. And if the nucleus is oval in shape like this, most likely the cell is columnar. But don't use this method unless you can't see the borders for you to identify. All right, so if you wanted to keep looking only at this one, I would say, yes, this is a cuboidal cell because it has a circular nucleus. All right, also you can see the thickness of the layer itself. Looking at this one, so you see here, I can't use this method this time. Why I can't use it? It has circular nucleus, but you can see definitely the clear borders of the cell. So don't rely on checking the shape of the nucleus to determine the shape of the cell unless you can't see the borders of the cell. So let's try this one. Do I see space lined with cells? What do you think? Yes, so it's epithelium. And do I have one cell layer or more than one cell layer? What do you think? I have one or more than one? One cell layer or more than one cell layer? Again, when we say more than one cell layer, the cells need to be on top of one another. So you've got a layer on top of another. If I do have the space between the cell layers, this tells me that those are not cell layers. Those are one layer on one side and one layer on another side. And you do have the space in between. So what we're looking at in here is actually a tube that I'm looking on the side of it. So I cut it in half and I'm looking at the side. So you got one row of cells lining the top, one row of cells lining the bottom. So having one cell layer, what's gonna be the type of epithelium? Is it gonna be simple or stratified? Simple. And what is the next factors that I need to check if I'm identifying the epithelium? How would I be identifying the epithelium other than the number of cell layers? Shape of the apical cells. And as I have one cell layer, the apical cell is the basal cell. So there is no difference. So we're gonna check here the shape of the cell. What do you think? When you look at the shape of the cells, look at one cell at a time to not get confused. Look at one cell at a time. So focus on one cell, look at it, and then move to another cell. Don't just look holistically. All right, so looking here at simple and the shape of the cells here, they are tall cells, so this is going to be a simple columnar epithelium.